Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Thank you very much for joining me for this talk today. As it is clear from this title slide here, I represent Computational Biology Service Unit, which is one of the core facilities at Cornell University. See if I got it to work. That's better. The mission of our unit is to provide the widely understood computational support and research support for biological research at Cornell University. In particular, we maintain several Windows-based compute clusters, which we make available to Cornell community, but actually our influences, so to say, reach far beyond Cornell, and we have a lot of users and hopefully a lot of friends also all over the world. And one thing that we learned from this computational support that we're providing is that convenience of access to HPC resources is the major issue in biology. The fact is that HPC is badly needed in biological sciences, but really researchers would rather spend their time doing their research rather than figuring out how to submit a job or write a script or stuff like this. I guess that's the statement that we've heard a lot during that conference. So our approach to solving this problem of convenience of access to HPC resources has been a web-based user interface to our clusters. This has been developed at CBSU for a number of years and has been working pretty well. Essentially, this is a web interface written in ASP.NET consisting of a bunch of submission pages which the users can use to submit different kinds of applications to our clusters. So we have about 30 applications in total. Here is an example of such a submission page for BLAST, which is parallelized. And we have also other applications in different categories, including population genetics, which is now becoming very popular. So we have a lot of programs in population genetics. So what the user has to do is to just fill out this form, provide all the necessary input files and set some parameters, and then click the submit button, which unfortunately didn't make it to this slide. And our interface, the code behind such a page, takes care of everything else. And we have this interface which is called BioHPC. We have this described in a poster. The poster is very easy to spot. It's all green, so you can say that in a way it really stands out. So please come and visit. While the web interface has been working very well for us, we have been pondering the question for some time whether or not to expose our applications as web services. This would have some impact for our users. For example, it would allow them to incorporate our applications directly in their analysis pipelines. And another advantage would be that we would get rid of the limitation of a web form as a user interface. In particular, jobs could be submitted and data could be retrieved from a client of choice. For example, a lot of biologists use Excel as a tool to store and organize and analyze their data. So it would be really nice to be able to submit HPC tasks directly from Excel and then retrieve data and analyze them also in Excel. And while we have been pondering this question, we came across a relatively new product from Microsoft HPC team. The product is called Computational Finance Server, and as the name suggests, it has been designed mostly for computational finance applications. However, it is in fact a pretty open-ended general tool for deployment and execution of HPC tasks. It is web service based, and it features Excel 2007 as the user interface. So it is sort of something what we have been looking for. So we decided as a proof of concept and feasibility test to adapt a few of our popular computational biology applications to CompFin. And CompFin is the short name for this computational finance server. So to start with, we adapted a program called Structure, which is one of the most popular pieces of software in the community of population geneticists, at least according to the statistics collected through our web interface. So here's the outline of my talk. First, I'm going to talk a little bit about what Structure is and what it does. Then I'm going to highlight some points about CompFin, and I'm going to describe our CompFin implementation of Structure. So, what is Structure? The objective of the program is to split a group of individuals into populations or clusters based on the known genetic characteristics of these individuals. So it's essentially a clustering problem. However, it's a little bit different from what we usually think about clustering. Usually there's some distance involved in that. Now here, we don't have distance. Instead, we have a statistical model consisting of several components. So first of all, we have, I don't think this works well. First of all, we have the set of our genomic data, which we called collectively denoted by X. In short, these are alleles at several loci for a set of individuals. I come back to the structure of this data set in a minute. Then we have a number of populations that we want the program to try to split this set of individuals into. And we have our model variable. So these things are fixed. And now we have our model variables, which are both multidimensional vectors. We have the vector Z collecting assignment of individuals to populations, and we have the vector P, which collects allele frequencies within these populations. And the model gives us the prescription to calculate the probability, conditional probability, of obtaining experimental result X, provided that P and Z are what they are. So what Structure does, it is trying to obtain sort of a best fit, that the P and Z, which provides sort of a best fit to this data. And of course, there may be many definitions of what we consider to be the best fit. One obvious choice would be to simply maximize this probability with respect to P and Z, which would boil down to a maximum likelihood method. What Structure does is a little bit different. It uses the approach based on Bayes' theorem to calculate the posterior probability distribution of P and Z given the data. So to do this, we need to assume some suitable priors on P and Z. The method that the program uses to estimate this posterior probability distribution is Markov chain Monte Carlo simulation, which essentially consists of generating a long chain of different Z and P combinations. It jumps from one to the other according to some acceptance rules and generates a lot of them and can go into thousands or millions or hundreds of millions depending on the particular need. And once this is done, this creates a sample of Z and P, or if you wish, a multidimensional histogram, which is an estimate of this probability in question. And once we have this estimate, we can do whatever people usually do with probability distributions. We can generate some summary statistics, averages, variances, confidence intervals, and so on and so forth. So in particular, primary output from this program is a table of inferred ancestry of individuals, which is essentially a list of probabilities of each individual belonging to each population. And this is, roughly speaking, average of the vector Z over that distribution. And another component of output is inferred allele frequencies within populations. And this is, roughly speaking, an average of the vector P. There are also some other output parameters, which I don't want to talk about here. What I wanted to point out is something a little bit on the technical node. Structure is something that would be called at this conference a legacy code. This is something written in C probably, definitely not in C#, downloaded from a developer's website as a free code. It has input it takes input in the form of text files and also spits the output in the form of text files. This is something, it's definitely a legacy code, something that would be called a legacy code. And this is something that we are trying to squeeze into this CompFin environment. Now, an important thing to realize is that for a given genomic data set X, we need to do multiple independent runs of structure. First of all, we need to figure out which number of populations is the best, because we don't know it in advance, so we need to make a lot of runs with different numbers of populations. Even with the same number of population, we have to make sure that our results are consistent, because this is, after all, a Monte Carlo simulation, which can go astray sometimes. It can get trapped in local maxima and provide unreliable results. So we need to repeat them a couple of times at least to make sure that we are on the same page with all the runs. Finally, we want to try sometimes different control parameters that control this MCMC process. Each of such multiple simulations is pretty long, can take hours to days, and therefore structure could really benefit from the compute cluster. It's an HPC task, and benefit from the compute cluster. We can run each of these independent simulations separately on a separate processor. And given the form of input and output, it could also benefit from Excel user interface. For example, these text output files, they are kind of itching to be imported into Excel and visualized somehow or analyzed otherwise. So that's structure. So now what is this computational finance server? So first of all, it's a set of functions, an API, a programmer's interface, which abstracts from specific implementation details of things like job scheduler or storage. Then, so this is something that lets us interact with the whole environment. Next, there is a web service layer, which is used to submit and monitor jobs and also eventually to retrieve the output data. There is a program called TaskPane, an Excel add-in, which essentially makes Excel a client to consume these web services. The Microsoft SharePoint server is used as like a meeting point to store the Excel templates and actual binaries of the program that we are trying to deploy. There is Microsoft SQL server, which is used as the data storage. Of course, we have to have a cluster running Windows Server 2008, and also I know that initially the whole system was running on the previous version of the Microsoft cluster software. And finally, last but not least, since this is a computational finance server, it supports a hefty SQL database of historical market data, which we are not going to use here as computational biologists. We are not that interested in historical market data. So we have a bunch of Microsoft productivity tools glued together to provide a uniform platform for deployment and execution of tasks on compute cluster. So to make it a little bit more clear, let us consider a question: what does it take to deploy a CompFin application? So first of all, if we want to deploy a CompFin application, we need to create an Excel template with some cells or tables within it designated to be input cells. They will hold our input data. And likewise, some other set of cells will hold output data. And since in the spirit of web services, this Excel workbook will be talking XML to the rest of the CompFin environment, these tables have to be mapped onto certain XML schemas. So we need to define the XML schemas which we think will describe our model best. And this mapping can be done in a very standard way within Excel. Excel has this XML Map feature which can be used here. So another thing that we need to prepare is a little piece of C# code, which is actually going to interface our program with the CompFin environment. So it's going to accept the XML input from Excel, and it's going to transfer, deserialize it into the C# structures, data contracts. Then based on this input, it's going to decide how many independent structure runs we want to run, and will launch, queue up appropriate tasks. Now, each of these tasks will be doing the actual number crunching. As I said, the number crunching is done by a separate executable, which we have here as a part of our system. So each of these tasks will have to create the required input text files, launch the executable, then parse the outputs, load them into appropriate C# structures, result data contracts, and then finally output them as XML from the program. Now, all these things, the input-output handling and also the launching of tasks, which obviously requires some sort of interaction with the job scheduler, these are taken care of by the API. So this is pretty transparent to a programmer. We don't have to worry about how the scheduler works or how to serialize or deserialize the data. Everything is taken care of by pretty simple to call API functions. So finally, the output data will be stored in the storage, which is located somewhere close to the cluster. And there's a lot of implementations possible for this result storage. What is implemented right now is storage in terms of SQL tables. So basically, all this XML is loaded into SQL as SQL tables and stored there until it is retrieved by clicking a button inside Excel through the appropriate web service. So finally, while these two components of our implementation are done, the Excel workbook and this C# wrapper code, we upload both of these components into the dedicated SharePoint site, which is going to make it possible for other users to retrieve it and run it. So the bird's eye view of the whole system, the way it looks from the user's perspective is this. So we have our user with the laptop. What he needs to do first is to fire up the Internet Explorer and contact the SharePoint site in order to retrieve the Excel template where he's going to supply the input. So this Excel template opens up on the user's machine. He supplies the input, and after that, he saves it back into the back into the SharePoint site, which is serving also as kind of a job repository where all these Excel workbooks corresponding to different jobs are being stored. And once the workbook is saved, the user clicks a submit button in the Excel, which calls a job launching web service and triggers all kinds of complicated events here, which eventually lead to launching of the whole job. So all this complicated part is completely hidden from the user. Of course, the only thing that he has to worry about is this part. And then once the job is complete, the user gets notified, and then he clicks another button in order to retrieve the data from the XML SQL storage. And there's another web service running here that takes care of that. So let's see what does it look like in practice. So first, we log into the SharePoint site. That's what it looks like, and we open our My Models library link, it's called, from where we can retrieve a template of our structure job. And this template, once we do that, the template opens up in Excel on our local machine, and it opens up with a welcome message containing some instructions for the first-time users, specifying how to use the program. And the next thing we need to do is to supply the data. So this is the actual genomic data, sample genomic data, denoted by X. And, well, what it is, we can see that, okay, so the first column is the identifier of an individual. So we have a bunch of individuals here. The first one is called 1, the second one is called 10, the third one is called 12, 29, I think, something like that. Then the second column, and we have like 155 of them. Not all of them are shown. The second column stands for some other characteristics of this individual, like, for example, geographic location here. And the rest of columns, they contain identifiers of the alleles found at the number of loci. So for here, for example, we have 7 loci, L1 through L7. And these integers here, they may have some physical meaning, for example, when we deal with microsatellite data. But other than that, they are just labels, pretty much. Now, each individual is deployed, which means that it has 2 instances of each locus. So that's why we have 2 lines per each individual. And for instance, if we look at individual number 10 and locus 1, we see that the alleles at this locus are the same. We say that this individual is homozygous at locus 1. On the other hand, if we look at the same individual locus 5, for example, we see that the 2 alleles are different, which makes this individual a heterozygote in this locus. So this is pretty much the type of input data that structure accepts, and that's pretty much pretty well structured and suitable to be stored and processed in Excel. So once this is ready, we just press this button here, which activates a macro, which in turn transfers this to the appropriately XML mapped portion of the workbook. So the genomic data is there. So what we need to do now is specify other run parameters. So this looks a little bit cryptic for now, but all the researchers who actually use structure in their research would know what all these parameters mean. For instance, we have the number of individuals specified here. We have the ploidity of them. We have other parameters. The most important part of this input is probably this. We have 15 lines here. We can have as many lines as we possibly want, and each of the lines corresponds to one individual structure run that we want to execute. As I said before, we need to execute several runs for each number of populations k. So for example, here we are scanning numbers of populations from 1 to 5, and we are doing 3 runs for each of these numbers. We can also specify a number of steps of the actual simulation and burn-in. We can do it separately for each run, but here actually they happen to be the same. So basically this table tells the program to execute 15 separate structure runs with these parameters, and the other parameters will be common to all these runs. So once all the data is there, the next thing we need to do is we need to save this workbook back into the SharePoint site, okay, using the Save button, and then once this is done, we just submit the job by clicking Submit. And when we do that, actually, by the way, this piece of Excel window here that we see is the task pane. That's this web service client, which is an Excel add-in. It has to be installed in Excel if you want to use it as the interface to Compfin. So it has many functions, and one of them is just submitting the job that we just did. While the job is running, we can monitor its progress using the standard progress bar, and when the job is done, we get notified that the job just completed. We do not have to keep the Excel workbook open all the time. We can just go for coffee or go on vacation, depending on the duration of the computations that we are doing. If we go for coffee and close the workbook in the meantime, can always come back to it, reopen it again, and we will have this notification waiting for us. So once the job completes, we are ready to retrieve the data. And there is a special button here in the task pane that does it for us. We click this button, and the data is streamed into the XML mapped designated areas designated for output. So this is an example of these areas. We have a special worksheet here that collects all these XML maps. And you can see that, XML map is mildly human readable. If you really want to, you can extract some information from it just by looking at it, but there is a better way to do this, and this way is based on standard Excel mechanisms, pivot tables, pivot charts, and also Visual Basic macros. So this is what we do. We basically, the table that you just saw, we process it through the pivot table mechanism, and this is what we obtain here. So this is essentially the record of all our 15, the summary of all our 15 runs, and some basic results from all these simulations are listed here. In particular, we have the number of populations, which was the input actually, and we have this logarithm probability of data, which is actually a very important number, because it tells us, it allows us to tell which population number is the best. For example, we can see here that for 3 populations, this number is the least negative, except for this middle result here. But other 2 are the least negative of all, and that's basically the indicative that that there's 3 populations, that the data implies 3 populations. Now, how about this point that sticks out? Well, as I said, the Monte Carlo simulation sometimes can go astray, sometimes not equilibrated too well, and this is an example of such occurrence. One way of analyzing the results of the run is to monitor the parameters which are sampled during the run. So one way of monitoring them is to make a histogram out of them. And we can make a histogram of any parameters. This actually opens up as a drop-down list from which we can select which parameter to view. So for example, here we have a parameter called alpha, which is, has a distribution centered around a pretty small number, much less than 1. And the theory tells us that this means that individuals are mostly purely from one population or the other. There is no genetic, not too much at least, genetic admixture between different populations in individuals. So this is the kind of information that we can get at our fingertips out of this. Again, we can make a lot of plots. This is actually a pivot chart, which is created out of another pivot table, which illustrates the allele frequencies here at locus 2. For example, we can see that allele number 2 is most abundant one in populations 1 and 2. This plot summarizes the ancestry of individuals. Basically, each individual is represented by a point located somewhere inside an equilateral triangle. And if this point is very close to one of the corners of the triangle, each corner corresponds to a different population. So if an individual is in the corner, it belongs to this population. At least this is what the genetic composition of this individual tells us. So here, and every individual also has a shape and the color associated with it, which correspond to some different prior characteristics of individuals. In this case, this is a location when the given individual was caught. And we can see that usually, at least in this example, the geographical location sort of corresponds to the genetically derived populations. However, there are some outliers. For instance, this guy here, he has the genetic composition of population 2, but it was found in the area where the population 1, sorry, but it was found at the area where the population lives, population 2 lives. So this kind of course, invites all kinds of scientific questions, and this information is also available here at our fingertips through Excel. And last but not least, for those who would prefer to have raw text output from the program, this is available as well, which can be this can be just exported from Excel using standard export mechanism. So what is the verdict? Is COMPFIN a good platform for computational biology applications? Well, certainly powerful Excel interface helps very much. We can do a lot more with this interface than with any other stand-alone graphical application which could analyze the data from this particular program. It's relatively easy to deploy to the COMPFIN environment. There's not too much programming involved in this. And finally, Confin offers this on-site data storage, which we have not used here because we didn't have such a need. However, this is something really to consider in view of the very data-intensive applications, such as next-generation sequencing. In fact, Confin has been developed with this idea of bringing computational power to the data rather than data to computational power, and that looks like something which would be useful for computational biology, when we, for example, envision Confin installation somewhere close to a sequencer which generates huge amounts of data which then have to be analyzed on an HPC system. There are a few things that could be improved to make Confin a better platform for computational biology. For example, currently all the input and output data transfer is exclusively through Excel. So this is very nice, but what if we have some binary data to transfer, or what if our data is not really Excelable, it's not pretty enough or not informative enough to be put into Excel, or what if it has 20 gigabytes, for instance? Also, there are some issues with the transfer of output from the SQL storage to Excel through XML maps. This may not be too efficient for very large data sets. I'm sure it can be improved more. Actually, it has been improved greatly as the result of this project, but it is a concern. And another thing that is a little bit, I would say, annoying from our point of view is that in order to use Confin, the user needs a domain account on the cluster. So this seems to be good for small, closed organizations, corporate organizations, for instance, but not so much for an open university research environment. For example, in our BioHPC web-based user interface, the number of independent users that actually utilize it is, what, a couple of thousand. Now, our clusters live in wider environment provided by our departmental CIT people, and they would be really upset if we asked them to suddenly set up 2,000 domain accounts just to use the system. So some sort of way to allow guest users would be really useful to be programmed into this system. So finally, let me acknowledge financial support from Microsoft HPC Institute program that we've been a part of for a number of years, and more recently from Microsoft Research, as well as fruitful collaboration with the Microsoft HPC team, in particular Richard Sepela and Daniel Simon. Thank you for your attention.